Let me introduce Stefan again. Stefan is a PhD student in my lab, the biomedical informatics lab. He completed his bachelor in mathematics at New York University. Shortly after that, he joined my lab at Memorial Sloan Kettering Cancer Center as an analyst, where he contributed to large cancer genomics consortia. Stefan then completed his master in computer science at ETH in 2019, and he's currently interested in machine learning approaches to modeling single-cell datasets. Today, he talks about SCIM, a universal single-cell matching with unpaired feature sets. Please, Stefan.
Okay, thank you, Gunnar. And thanks to the organizers for the opportunity to present. So today I'll be talking about a framework, SCIM, that we've been working on to integrate multi-omic single-cell datasets. This is joint work with Joanna Ficek from the Biomedical Informatics Lab. So single-cell technologies are a recently developed class of methods that are able to profile samples at the cellular level. So consider a heterogeneous sample of cells. The sample contains cells that exist in several different states. And these states correspond to something like cell type or progress in a developmental process. And here we're going to represent cell state using these colors. So if you were to profile this sample using traditional bulk methods, then a single measurement would be taken. And this measurement's a joint signal across all cells. And the problem with this is that this sample is composed of cells in a distribution of states, and this measurement doesn't explicitly capture this. But single-cell technologies, which profile cells individually, can explicitly capture these signals. So in this cartoon, the single-cell technology reveals that each cell state is carrying its own signature in feature space. And these signatures are represented using the colored histograms. And the data generated by these technologies, it can often be represented as a matrix of cells by features. So here, each row in this data set corresponds to a single cell in the original sample. And these technologies have generated a lot of excitement and have advanced fields by providing data that's better suited to study complex phenomena, such as cellular development and tumor heterogeneity. So there's currently many different single-cell technologies that exist, and these are able to measure cell states across some omic spectrum. So for example, there's technologies for genomic and transcriptomic and proteomic measurements, and there's imaging technologies that can measure samples on a spatial level. And these measurements that these technologies are taking represent these kind of unique but incomplete perspectives on the cell. And so if you were to somehow integrate these perspectives, you would generate a more holistic view of the cell and its environment. And this would allow for analysis that would not have been previously possible. So you could consider an experiment, for example, where you profile cells using both single-cell RNA-seq and IMC technologies. So the RNA-seq technology measures the gene expression within a cell across thousands of genes. And you can use this information to compute or to study like some complex underlying mechanism, such as pathway activity. And meanwhile, the IMC technology images cells stained by a handful of markers, of protein markers. And so this technology captures spatial relationships, but the small number of markers that it profiles makes it not possible to compute something like pathway activity. But if you were to integrate these two data sets, then the relationships between the pathway activity that you've got from the RNA-seq technology and the spatial effects that you get from the IMC technology can be studied. So here, an IMC image is recolored using hypoxia activation computed on the RNA-seq measurements. So an integration of this type, it's in general challenging since most technologies consume their sample. So you can imagine if you somehow profiled the same exact sample using two technologies, then you would generate two data sets with the same sets of cells in it. And because the technology destroyed this input sample, you don't know how the cells from one data set are related to the cells in another data set. So to integrate these data sets, we introduced the SCIM framework. SCIM stands for single cell integration via matching. It assumes that a sample is distributed uniformly to two or more technologies, and that these technologies consume their inputs. These technologies make datasets that can be represented as a cell by feature matrix, and we integrate these datasets by identifying cell pairs across the technologies. So crucially, SCIM does not require that technologies profile across the same set of features. So for example, if one technology measures gene expression, another can measure protein levels, and the protein's profile do not need to correspond to any single gene profiled. It scales to the large datasets that these technologies can generate. These can often be on the order of hundreds of thousands of cells, and it can integrate two or more technologies. So since each technology receives a similar distribution of cells, SCIM assumes that the underlying structure, which is here represented by the color clouds, is shared between all datasets. This assumption is key, and it's what allows SCIM to integrate datasets without requiring shared features. Integration proceeds in two steps. In the first step, the shared underlying structure is reconstructed and aligned within a low-dimensional latent space that's invariant to the technology. This means that similar cells profiled by different technologies are placed in close proximity, while the distribution in the space reflects the underlying structure in the sample. So to construct this space, we use a variational autoencoder framework introduced by Professor Euler. This framework has an adversarial component in the loss function to encourage the alignment of each technology's distribution. It aims to find low-dimensional representations of cells that retain information that's specific to the cell state, but that have lost all information specific to the technology. So the way this is set up is that encoder and decoder networks are assigned to each technology, and you can tailor their architectures to the specifics of their respective data types. And these networks operate as they would in a normal variational autoencoder. So this means that the encoder maps input data to some low-dimensional space. An embedding is sampled from this in this low-dimensional space from the encoder, and the decoder tries to reconstruct the input from this embedding. And these networks are trained together to minimize the reconstruction error. So data is embedded into this latent space, this low-dimensional latent space, and then the decoder tries to reconstruct the original signal. And if you were to do this for each technology that you have, you would produce embeddings or representations that accurately reflect the state of the cell. But there's nothing here that's imposing technology invariance. So you could embed cells from different technologies in different parts of the space or in an arbitrary orientation. And from this means that you can identify the technology of the cell from the embedding. So to impose technology invariance, a single discriminator phi is trained simultaneously to identify the technology of the embeddings. So that is, phi is trying to discriminate between the embeddings from one technology from the embeddings from all other technologies. And technology invariance is imposed by training the encoder to fool the discriminator. And this is an example of an adversarial model. So by fooling the discriminator, the distribution of each technology's embeddings become aligned in this latent space. And so when trained successfully, SCIM produces embeddings that are well integrated across technologies and reflect the underlying structure of the original sample. And SCIM is able to scale to large data sets through the use of gradient-based optimization. So SCIM is an adversarial model. And training these models can be difficult. So to help train this, we address two major challenges in the optimization. So the first challenge we address is correctly aligning the substructure inside the latent space. So you could consider, for example, that we wish to align a single technology to the substructure defined by these colors. So you could produce embeddings represented by these gray hexagons. And these are gray since SCIM is not explicitly aware of how they should relate to this background distribution as we've described it yet. And from inspection of these embeddings, everything seems to be well behaved. So the decoder can correctly reconstruct the inputs, and the discriminator is fooled since these embeddings are aligned to the underlying background distribution. Well, let's say after matching is performed, it's revealed that the blue cells were matched to the red cells and the red to the blue. And this can happen because the encoder can map the red cells to the blue space and the blue cells to the red space, and then the decoder can reverse this mapping. And this problem arises naturally due to symmetries in the latent space. So for example, the blue cloud has the same relationship to the green and orange cloud as the red cloud does have to the green and orange. And so this arbitrary switching is natural here. So to overcome this problem, we break this symmetry and we help it orient the latent space by providing the discriminator with categorical labels that inform it about the underlying structure. And so these labels could be cell type, for example, but in general, it's just something that captures some information about what's shared between these two technologies. And so to supervise, the discriminator inputs are concatenated with one-hot representations of these labels. So the discriminator sees the latent encoding, so the latent embedding, and then a code that describes the label. In most applications, the labels for a small subset of cells are enough to successfully orient this latent space between the technologies. So in this semi-supervised setting, cells that are labeled act as anchors, and these will define and coordinate the space around them across all technologies. So the second challenge we address is model selection under adversarial training. And this is in general not straightforward. So in adversarial training, the practitioner is not afforded some value, like the negative log likelihood, that can be used to select the best parameter settings. And so tuning a model becomes difficult. And often you need to use qualitative inspection or domain-specific metrics to measure how your model is performing. So in SCIM, we use a universal divergence estimator to assess the alignment of embeddings between a pair of technologies. So this estimates the symmetric KL divergence between two distributions using only samples from from these distributions. And this provides the user with a measure of how well SCIM is aligned the latent space between a pair of technologies. And it can do this with just a set of embeddings. So for example here, the light blue and the dark blue points could be the embeddings from two different technologies. And as these embeddings become more aligned or overlapped, the divergence estimate approaches 0. And if you have this, some measure like this, then you can use it in conjunction with the reconstruction error. And this can help model selection and parameter tuning by identifying successful runs at scale. So once the technology-invariant latent space is constructed, cells from all technologies are embedded into it. And then in the second step, the integration proceeds by pairing similar cells across the different technologies using these embeddings. So cell-to-cell matching, unlike a generative approach using the decoder networks, will constrain the matches to the true data distribution. And by pairing a measurement in one data set to a measurement in another data set, you can reduce downstream errors due to artifacts that you could inadvertently introduce using a generative approach. So you're basically guaranteed to have a realistic pairing or measurement. So SCIM performs this cell-to-cell matching between technologies pairwise. And it operates in the low-dimensional space, and it uses a fast bipartite matching algorithm using a min-cost max-flow approach. So the data from two technologies, here we're representing it as circles and hexagons. We can represent this as a bipartite graph, where each node is a cell embedding. And to scale to large data sets, we constrain the inter-technology edges to the k nearest neighbors of each cell. So these edges are weighted by the Euclidean distance computed on the low-dimensional embeddings. And to account for outliers and statistical differences in the distribution of cells between the two technologies, we introduce a null node, and this allows for non-matched cells. So this node is connected to all cells in the root technology, and it's given a large cost and infinite capacity, and this allows for but penalizes any unmatched cells. And then matching is performed in the usual way by computing the maximum flow through this graph and then minimizing the cost of the flow. And since these edges were computed using the Euclidean distances, you're more likely to pair yourself to a cell embedding that's close to you in latent space. So we investigated the performance of SCIM on real-world and synthetic data sets. So first, we tested SCIM's ability to integrate substructure across multiple technologies using simulated data with the PROS tool. So this tool PROST simulates single-cell datasets by parameterizing a negative binomial distribution with an underlying temporal branching process. And a hierarchical structure like this is commonly found in single-cell datasets. So this parameterization is continuous through this pseudotime. So each branch has some distinct signature, but these signatures evolve continuously in this pseudotime between branches. So you can see this as mimicking a developmental process. So cells could start in some state A, and as time or pseudotime evolves, you develop from A into branch C, or from A into B, likewise from B to D or E. And so PROST generates cells, and in each of these cells will have a branch label and a pseudotime label. And then, so a single tree is defined, this tree here, and we use this tree to generate 3 datasets. Each dataset is rather large with 64,000 cells and 250 features. And now the parameters from each dataset's negative binomial distribution are generated using different seeds. So this means that the 3 datasets that we consider, they have the same underlying structure that's defined by this tree, yet each of their feature sets are following different parameterizations, so you can't directly compare features between technologies. So we choose one dataset as a source technology, and we initialize the latent space by training a VAE. Then we run SCIM to embed and align all technologies into the shared latent space, supervising using the branch label. We visualize this space by computing t-SNE projections on the joint set of embeddings. So each column here shows the embeddings from a single technology, and these rows are colored by the branch label and the pseudotime. And it can be seen that the structure is reflecting the structure defined by the tree, and it's aligned across all 3 technologies. So for example, here, the blue cells that are in branch A are neighbored to the green cells from branch C and the orange from branch B. And this relationship is preserved across the different technologies. And if you look in the temporal coloring, interpolations in this latent space are reflective of paths in the tree. So for example, if I were to start with a cell in branch A and move into branch C, then this movement is tracing a path in this tree. And this relationship is generally followed by the embeddings. So this is the first step of SCIM to produce this integrated latent space. And then next we investigate the quality of the matches made by SCIM in its second step. So here we show a density plot that shows the alignment of pseudotime information across matched cells between a source and target technology. So this is computed over a scatter plot where each point represented the pseudotime pair, the pseudotime values of a single match. So correctly matched pairings with correctly matched branches are colored by the branch label. And incorrectly matched branches are shown in gray. And you can see that these are correlating with the bifurcation points in the graph. And here the labels are ambiguous because the way that PROST goes between branches, this transition is happening in a continuous manner. So the label here is ambiguous. And so we quantitatively evaluate the matches that SCIM's made, and we show that it's making good matches, preserving the branch label in 86% of matches, and preserving pseudotime values with the Pearson correlation of 0.86. So most of these matches are on the diagonal. We've also applied SCIM to a melanoma sample profiled by single-cell-seq and CyTOF technologies. And this yielded a data set of around 140,000 cells. So here we're showing a t-SNE projection of the integrated latent space that SCIM produced. And we show that SCIM correctly aligns this latent space between the technologies using a semi-supervised strategy with 10% of the cell labels. So that means 90% of cell labels were removed, and it was still able to integrate. And it produced matches of high quality, retaining cell type in 93% of matches. So in conclusion, we've developed SCIM, a tool to restore lost cell correspondences in multi-omic datasets. SCIM is short for single cell integration via matching, and it proceeds in 2 steps. First, it constructs low-dimensional representations of cells that are invariant to the technology. Then in the second step, it matches these representations using a fast bipartite matching scheme between technologies. It doesn't require feature correspondences between datasets. It's applicable to any embeddable data type, and it scales to datasets with over 100,000 cells. SCIM can integrate multiple technologies at once, and while its adversarial training can be difficult, it addresses challenges to its optimization. So for more details, we have a manuscript available on the bioRxiv that's accessible through this link or QR code. I'd like to thank my collaborators, especially my first co-author, Joanna Fitzek, as well as our supervisors, Gunnar Retsch and Kyung Lemon. And thank you for listening, and I think I can take some questions.